March 2015. It's a Saturday night. 5 teenagers are gathered in a parked car inside a gated community, when suddenly a police officer shows up and begins asking questions. The kids tell him that they're there for a sleepover, but the officer continues his line of questions and mentions that there have been recent car break-ins in the area. The kids are then surprised at the officer's next request. He asks, which of the 5 teenagers are willing to provide a DNA sample? Now let's pause right there, because at the inception of DNA profiling, the technology was reserved for cases involving serious violent crimes. Even then, investigators understood that the need for justice must be delicately balanced against the need for the medical privacy of our citizens. Their solution was to select regions of the human genome to create a genetic fingerprint system that identifies suspects with the specific requirement of not revealing any medical information. To create this fingerprint, they selected non-protein coding regions of the human genome that consist of repetitive sequences of the nucleotides that make up our DNA. These repetitive sequences are called short tandem repeats, or STRs for short, and their lengths vary from one individual to the next. An individual's combination of STR variants is used to create a genetic profile. That profile is then added to the FBI's Combined DNA Index System, CODIS, which now consists of tens of millions of genetic profiles used for forensic identification. Since its inception, law enforcement has increasingly expanded the use of DNA evidence. In some jurisdictions, even a chance police encounter like getting pulled over can result in the collection of a DNA sample. Since the development of CODIS, however, the Human Genome Project finished sequencing the entire human genome, and tons of genes have been discovered. These achievements helped open the door for what are known as genome-wide association studies, or GWAS for short. A GWAS is where researchers find associations between diseases or traits and genetic variants. After the first GWAS, hundreds of thousands of single nucleotide polymorphisms, or SNPs for short, were found to be associated with various traits and diseases. We'll refer to these as GWAS SNPs. These advancements in genetics compel us to reassess the assertion that CODIS STRs only provide identification information akin to a fingerprint. Previously, the Rohlfs lab at SFSU found some CODIS STRs to be close to or even within gene expression modulating regions and associated to gene expression. We've covered this in more detail in our previous video and publication, which are linked in the description. Then, Dr. Rohlfs' lab at SFSU and Dr. Edge's lab at USC collaborated to further investigate if CODIS STRs could be informative about GWAS SNPs. We found that, compared to other STRs in the genome, CODIS STRs are in the 97th percentile for the number of pathogenic variants found in close proximity. A striking example is TH01, which was identified by our lab and others to be associated with the genetic expression of a gene that plays an important role in synthesizing dopamine. TH01 is surrounded by an impressive 10 GWAS SNPs, which are associated with traits such as type 1 and type 2 diabetes. It's also surrounded by a whopping 25 pathogenic variants, each of which is associated with a disease like permanent neonatal diabetes or dystonic disorder. It is unclear exactly why CODIS STRs are in regions unusually saturated with these phenotypic associations. It may be that in order to sequence the CODIS profile, the DNA regions must be accessible for PCR amplification. This requirement may have unintentionally biased the selection of CODIS STRs for the regions that exhibit higher transcriptional activity, potentially enriching for CODIS STRs with functional relevance. However, we are still unsure. What we are sure of is that these findings directly challenge the outdated assertion that CODIS STRs may serve as genetic fingerprints devoid of medical relevance. Given the information you've learned today, are you convinced CODIS STR profiles are simply identifiers? Or do they potentially invade a person's medical privacy? And also, how do you think we should use DNA evidence? To learn more about this study, you can read it on iScience via the links in the description. We'd like to thank all of our funders as well as our amazing team for their groundbreaking research, and thank you for watching.